Okay, today we're going to talk about enzymes. And please remember that enzymes are proteins. That's the type of organic compound they are. So they are proteins that speed up chemical reactions by lowering something called the activation energy, the energy that's needed to get a chemical reaction started. So if enzymes speed up chemical reactions, we need to understand a little bit about what a chemical reaction is. So chemical reactions occur when chemical bonds, those are bonds that are holding atoms together, break or form. And when you look at a chemical equation like the one here, we've got two hydrogen peroxide molecules are going to be broken down into two waters, H2O, and two oxygen molecules, oxygen gas. So whatever is on, once you find the arrow, whatever is on the left-hand side of the arrow, those are going to be called reactants. So those are the reactants that are needed for the chemical reaction. What is ever on the right-hand side of the equation are going to be called products. So in this particular chemical equation, the hydrogen peroxide is the reactant, and then water and oxygen are the products. So let's answer the question, what is a catalyst? Okay, well, enzymes are examples of catalysts, but there can be others. So a catalyst is a substance, and I'm going to abbreviate substance as SUBS, that speeds up the rate of a chemical reaction. And I'm abbreviating reaction RXN. So again, I mentioned above something called activation energy. Okay, well again, activation energy is the energy needed to start a chemical reaction, the energy needed to start a chemical reaction. So there's a little bit of energy that's needed to get a chemical reaction started. And so enzymes are going to then speed up a chemical reaction by lowering the activation energy. So if you look at this graph, and I've highlighted it with using a yellow and a green highlighter, and you basically see two curves. And of course, on the y-axis is energy, and the x-axis is labeled, it just got cut off, but it's the progress of the reaction. So if we look at the progress of the reaction, and again, versus the energy, you can see that there's a certain amount of energy in the reactants. And let's look at the green line first. So the reactants have a certain amount of energy, and you can see that it's going to, the energy needed to go up a little bit, and then the products will form. Well, that little bump right there, that little increase right there, is something called the activation energy, the energy that's needed to get the chemical reaction started, so then the products can form. Okay, well, if you look at the yellow line, which is going to be the solid line, you can see that the bump is a lot higher. Okay, it goes up a lot, of it's needed more. Okay, again, one of these curves is showing when a reaction is using an enzyme, and one is showing you when the reaction is not using an enzyme, when an enzyme is not involved. Okay, so again, the yellow line is showing you the progress of the chemical reaction when there's no enzyme used. Okay, so the yellow, again, is no enzyme. The green is showing you when there is an enzyme used. Okay, so you can see that when there's an enzyme that's being used in the chemical reaction, the activation energy is much lower, okay, than when there's no enzyme used. So let's just pretend that you were running a race, and there were two courses. You had the yellow race, and then you had the green race. Okay, well, which one could you run at a faster pace? Obviously, the one in green. You have less of a hill to climb, so to speak. And so again, enzymes will speed up chemical reactions by lowering that activation energy. Now, when we look at how enzymes work, that is going to be something called the lock and key model. Okay, the lock and key model. And there's a few things that we need to remember about enzymes. Each protein has a specific shape to it. So enzymes can only bind to substrates that match its shape, kind of like that a key is going to unlock just a specific lock. My house key is not going to be able to unlock my car, for example. Okay, the site on the enzyme where the substrate actually hooks on is going to be called the active site. Okay, so we've got some pictures here. Let's look at this one first. So here is the enzyme, and you can see it has a very, very specific shape. And these indentations, those are going to be the active sites, because that's going to be the area that the substrate, this compound, this reactant, is going to bond to. So you've got these chemicals, substrates, they're reactants that have a certain shape, and you can see that the enzyme matches it, just like a lock and a key match. That's where it got its name from. And so again, the active site is going to be this area in here. Has a very specific active site. So for each enzyme is going to have a certain shape that fits a specific substrate. So when we look at the lock and key model, we're going to do some labeling. So A is going to be the substrate, some compound. B is going to be our enzyme. And it has an active site. Okay, so again, here in the yellow is the active site. That's where the active site is going to be. So when the enzyme and substrate come together, okay, C is going to be the enzyme substrate complex. And I'm abbreviating enzyme. So the chemical bonds and the substrate start to weaken, and you end up having products form. So E are products. So maybe this was hydrogen peroxide. And of course, it needed an enzyme. And then after the chemical reaction happens, you've got water and oxygen. So these would be products. Down here, D is the enzyme. Now, that's something that's very important. Notice that the shape of the enzyme did not change. So here's before the chemical reaction. Here's after the chemical reaction. The enzyme did not change. The shape of the enzyme did not change. So that means that enzymes can be reused over and over again. So this enzyme can now go and bond with another substrate and break down that compound. So enzymes are reusable. Now, how can we change how well an enzyme works? Okay, so enzymes can be reused. They are reusable. Now, there are certain environmental factors that can affect enzymes and how well they work. So enzymes are affected by temperature, specifically high temperatures, and pHs. Specifically, usually there are some exceptions. Most enzymes work best at around a neutral pH. Again, there's some exceptions, like the enzymes in your stomach can work in an acidic environment. But for the most part, they prefer a neutral environment. So what happens when the enzyme encounters a strong acid or a strong base, or maybe even high temperatures? Okay, that can change the shape of the enzyme. And if the shape of the enzyme changes, okay, then it's not going to be able to bond to the substrate and the chemical reaction will not be able to take place. So there's a term that describes when an enzyme changes its shape. Okay, again, it's been damaged in some way. Again, usually because of high temperatures or extreme pHs, strong acids and strong bases. And that word is denature. Okay, denature. When an enzyme breaks down or changes shape, that's not going to be good. That means that the chemical reaction cannot take place. So there are things called buffers out there, and we've talked about those before. Okay, buffers keep the pH stable. Okay, because again, most of the time, enzymes in your body does not like to have the pH of the blood, for example, to change. And so we have buffers, which are weak acids and weak bases that help keep that pH stabilized. Okay. Now a little bit more over here. What are some examples of enzymes? Well, if you remember, enzymes, again, usually end in ACE, A-S-E. So for example, catalase, there it is, catalase. Another name for it is peroxidase, is an enzyme that breaks down, okay, this is a mistake. It should not say starch there. It should say hydrogen peroxide. Which the chemical formula for hydrogen peroxide is H2O2. Okay, so catalase breaks down hydrogen peroxide. Lactase, there it is, there's the ACE again, will break down the sugar lactose. And lactose is a sugar. It's found in milk. It's a disaccharide. And so a lot of people that are lactose intolerant, their lactase is not very effective. And so they have trouble breaking down the sugar lactose. Amylase, again, there's the ACE. It's an enzyme that breaks down amylose, which is starch. Another way of saying starch. Again, remember, starch is a type of carbohydrate. Carbohydrates end in ose. And you have an enzyme called salivary amylase that helps to break down starch that's in your food once you immediately put it in your mouth, your saliva. The salivary amylase goes to work on it. So again, enzymes are proteins that speed up chemical reactions. They do so by lowering, look at the green, lowering the activation energy, the energy that's needed to get the chemical reaction started. When we look at a model of enzyme activity, it can be called a lock and key model. And enzymes have a very, very specific shape that will fit to a specific substrate or reactant. When they come together, it's called an enzyme substrate complex. The chemical bonds weaken, products are formed, and notice that the enzyme is unchanged so it can be reused over and over again. High temperatures and extreme pHs can denature enzymes, which means that they're going to change the shape, and that means that the chemical reaction cannot take place. Okay, so that's a little bit on enzymes. A little bit on enzymes. Again, this came from page 36 in your packet, and it's going to be put on page 45 of your notebook.